I probably won't need the microphone. I usually don't have any problem being loud, too loud. Okay, I'm going to be talking to you about an unappreciated potential source of variation in flowering phenology in Arabidopsis thaliana, involving methylation, of course. So Tomas did a really great job of setting me up in a lot of ways for some themes that you're going to see in my talk and probably a lot of talks throughout the day. But really quick, I'll give my definition of epigenetics. Phenomena that cause chromatin modifications and have the potential to affect gene function in the absence of changes to DNA sequences. So we're talking about changes in gene expression that are not underlain by nucleotide polymorphisms. Some examples of epigenetic mechanisms that are commonly studied are DNA methylation. This has to do with the, as Tomas set me up for, the addition of methyl groups to specific cytosine nucleotides within the DNA sequence. There's also RNA methylation. There's histone modifications, and there's also siRNAs that are all epigenetic mechanisms. But this talk is going to focus for the most part on DNA methylation. Again, very similar to Tomas's slide, in a very simple form, if you have I hit the wrong darn button. If you have a region upstream of a gene, for example, where a group of cytosines are methylated, this can repress the expression of that gene as compared to the situation where you have the exact same nucleotide sequence, but where you don't have methylation of the cytosines upstream of the gene and the gene is expressed. A little bit more into the mechanics of how methylation is achieved in Arabidopsis. You have this group of genes known as your Chromatin Remodelers, and well, they code for proteins that are chromatin remodelers, and the chromatin remodelers unwind and unpackage the DNA strands, making them accessible to these genes here, the methyltransferases, which actually add the methyl groups to the specific cytosines in the DNA sequence. So I want to talk to you about these DNA methyltransferases that actually add the cytosines, that actually add the methyl groups to the cytosines. In studies that have used mutant lines that have loss of function mutations for these DNA methyltransferase genes, there's some specific phenomena that they observed in all of these different loss of function mutants for these different methyltransferase genes. For example, when the function of this DNA methyltransferase, methyltransferase 1, is knocked out, this causes the genome to be hypomethylated, a greatly reduced amount of cytosine methylation genome-wide. And it also results in those plants being late flowering. Similarly, when DRM1 or DRM2, when the function of those genes is knocked out, so that they're not expressing that gene, they are also hypomethylated and late flowering as well. So that's the situation that you see in these mutant strains relative to the wild type from which they are derived. So just the mutant relative to the same genetic background in the wild type. But this is an evolution meeting. Here we're interested not in mutants relative to the wild type. We're interested in natural variation, because natural variation is the fuel of evolution. And Arabidopsis has lots of natural variation, genetically, phenotypically. It's a cosmopolitan species. Its native range is all throughout Eurasia. It also has a wide introduced range within North America. It occupies a wide variety of different latitudes, and it has a wide variety of different phenotypes that you see at in different locations throughout its range. So it's got all the ingredients for understanding natural variation. Okay, so I set the stage for you with what's going on in these mutant lines, these DNA methyltransferase mutant lines. Now I want to show you some data involving natural variation in Arabidopsis. And so the data I'm going to be showing you is going to come from two sources. It's It's going to come from this paper from Atwell et al. In 2010 from Nature. That's where the flowering phenology data that you're going to see comes from. And then also the data that you're going to see on gene expression levels and on amount of methylation and methylation patterning genome-wide comes from this Schmitz et al. Paper that was based upon whole genome bisulfite sequencing. In both cases here, we're talking about 100 different individuals of Arabidopsis thaliana that were collected from throughout its natural range in the wild. And as has been documented many times before, flowering time in Arabidopsis thaliana has a quantitative variation. So individuals vary in their flowering times following a roughly normal distribution. And it also turns out that individuals of Arabidopsis thaliana from throughout its natural range have quantitative variation in the number of methylated cytosines genome-wide. So we've got that ingredient in there as well. And if you look at the expression of DNA methyltransferase genes, individuals collected from the wild vary in a roughly quantitative fashion in the expression levels, in their constitutive expression levels of these methyltransferase genes. All right, in the mutants, no expression of a DNA methyltransferase gene leads to hypomethylation genome-wide, or reduction in methylation genome-wide, which causes delayed flowering. So that's the case in mutants. This is an evolution meeting. We're interested in natural variation. So the analogous slide, does natural variation in methyltransferase expression explain variation, natural variation among individuals in the number of methylated cytosines genome-wide? And does the number of methylated cytosines genome-wide explain natural variation in flowering time? So this is the analogous scenario with evolutionarily potential variation. Okay, let's focus on the first part of this question here. Does variation in methyltransferase expression explain natural variation in the number of methylated cytosines among individuals? And the answer appears to be no. So we have a mismatch between what you see in the mutants and what you see when you look across natural strains. So for example, with methyltransferase 1, with that DNA methyltransferase, as you increase in expression, there's no relationship to the number of methylated cytosines. And this is looking at individual strains plotted out here for this regression. There's no relation. Okay, let's look at the second part of this question. Does variation in the number of methylated cytosines explain some of the variation in flowering time among naturally collected strains? The answer again is no. So we have another mismatch between what you see in the mutant analyses and what you see when you look at natural variation. So that's strange. But take a look at this result here. There's another piece of data in the Schmitz et al. Data set that we looked at, and we found something really interesting. Okay, the y-axis here is still flowering time. Here on the x-axis is the number of single methylation polymorphisms that an individual has relative to a laboratory reference strain known as Columbia. And it turns out that the more different the patterns of methylation are of an individual as compared to this Columbia reference strain, the later it flowers. So we're not talking in this case about the amount of methylation. We're talking about dissimilarity. Either some places are methylated relative to Columbia, some places are unmethylated relative to Columbia. Dissimilarity relative to Columbia is associated with increasing flowering phenology. And so why would that be the case? Well, the Columbia strain is not the laboratory strain for random reasons. It's the laboratory strain of choice because it's easy to work with and it grows really fast. So here it is in terms of the distribution of flowering times. So if you have something that's an extreme early flower, this here is a genetic network summarizing the main things that we know about how flowering is achieved in Arabidopsis. And what I want you to see is that there's genes in this network that repress genes farther down in the network There's genes in this network that promote genes farther down in the network. So a wholesale addition of more methyl groups to genes in this network isn't going to accelerate or decelerate flowering. What's going to be important is what particular genes are methylated versus unmethylated. So the patterning is what's going to be important. So it makes sense that if there's a change in the patterning relative to Columbia, that this would generally serve to accelerate flowering. If things get even weirder, this is a protein methyltransferase. I don't know what to make of this, but we found that the level of expression of this particular methyltransferase is negatively correlated with dissimilarity to Columbia. So the greater the expression of this gene, the more similar the methylation patterning is to Columbia. Not more methylation, but just more similar methylation patterning. So, and we found other genes where there was the same pattern like I just showed you on this graph, where it seems that these protein methyltransferases and these RNA methyltransferases are somehow influencing the overall patterning of methylation genome-wide based upon their expression levels. So in summary, natural variation in methyltransferase expression influences genome-wide methylation differently than you see in methyltransferase mutants. And natural variation genome-wide in methylation influences flowering differently than the hypomethylation that you see in mutants. And so it seems that natural variation in genome-wide methylation influences flowering phenology in a complex manner. And then, even more interesting still, some methyltransferases have their expression associated with genome-wide cytosine methylation patterns, and not the amount. And so these methyltransferases may influence natural variation in flowering phenology via the patterning of methylation that they cause genome-wide. I also want to use this here as an advertisement. We have a symposium through the Society for the Study of Evolution tomorrow from 8:15 a.m. to noon on epigenetics and evolutionary processes. Bob Schmitz, whose work was featured prominently in my talk today, is one of our speakers tomorrow. We have a variety of speakers from around the world talking about different taxa and the relationship of epigenetics and evolutionary processes in those taxa. I hope many of you can make it. And with that, I'm happy to take any questions, if I have time. Yes, go on.
So in addition to looking for an association between natural flowering time and what's happening at these methyltransferases, have you looked at what's happening at some of these flowering time genes in the pathway?
Right, yeah, that's, I mean, we're early on in this, and we want to mine this data and look at those associations as well. Yeah. Really dig down deep and see if there's methylation patterns that are changing in specific genes that might be accounting for more of this variation. Yes?
Josh, when you show the correlation between methylation polymorphism different from Columbia and flowering time, you show that the greater distance you see that correlation. What if you just use SNPs?
Yeah, that would serve as a good control for population structure. Haven't tried that. That could potentially account for some of that variation as well. I suspect not all of it, but that's a good point as well.
Okay, we're going to have to move to the next speaker. Thank you very much, Joshua.